Welcome. Our topic is physiology of flowering plant. We come from SMK Srinivas Selatan and our group members are Brian, Amy, Yijun, Kianfeng, and Harish. We chose this topic because we are currently learning about the plants and are interested in their structures. This team explains basic plant processes, including tissues, growth, leaf structures, transport, and plant adaptation in different habitats.
Organization of plant tissues. A plant contains of two types of tissues, which is meristematic tissues and permanent tissues. What are both of these? Let's talk about meristematic tissues first. Meristematic tissues are the actively dividing tissues through the mitosis. There are two types of meristematic tissues, which are apical meristematic tissues and lateral meristematic tissues. What about permanent tissues? The permanent tissue contains of epidermal tissues, ground tissues, and also vascular tissues. The permanent tissues are matured tissues that have undergone differentiation. Epidermal tissues. Epidermal tissues are the layer of the outermost surface of the plant. The epidermal cell walls have a waxy waterproof layer which is called the cuticle. The cuticle protects the leaf from mechanical injuries and reduces the loss of water through evaporation, which is also known as transpiration. These are the example of the cuticle on the leaf. Modified epidermal cells and their functions. The guard cells are used to control the opening of the stoma, while the root hair cells are used to increase the surface area of the root to absorb water and mineral salts. Next, let's talk about ground tissues, which are under permanent tissues. Ground tissues are separated to three types. Parenchyma tissues are the simplest living cells without undergoing differentiation. It contains thinner cell walls and are always in turgid state, providing support and the shape for herbaceous plants. Parenchyma tissues are involved in photosynthesis and gaseous exchange. It helps in the storage of starch and sugar. It is also involved in repair and regeneration of plant tissues in the vascular system. Parenchyma tissues. It is made of living cells that are matured into flexible cells. It contains cell walls that are made of pectin and hemicellulose. Parenchyma tissues have thicker cell walls than parenchyma tissues, and it provides mechanical support and elasticity to plants. Sclerenchyma tissues. It is made of dead cells at maturity. It has the thickest cell walls among ground tissues and provides support and mechanical strength to mature plants. It also helps in transporting water and nutrients. Now, let's talk about the vascular tissues, which consist of xylem and phloem. The xylem is made out of dead cells and its cell wall contains lignin, which provides rigidity. It is to transport water and mineral salts from roots to all parts of plant. However, the phloem is made out of living cells, which is the sieve tubes. It is to transport organic substance from leaves to all parts of plant.
There are two types of meristematic tissues, which is apical meristems and lateral meristems. Apical meristems are at the tips of roots and shoots for primary growth, while the lateral meristems are along stems and roots for secondary growth. Three zones of cell growth at the tips of the shoots and roots of plants, which is zone of cell division, zone of cell elongation, and zone of cell differentiation. First, the zone of cell division takes place at the apical meristems, where meristem cells are actively dividing by mitosis. Cell number increases and elongates the plant stem. When new cells are forming, the cells formed previously are pushed to the zone of cell elongation. The zone of cell elongation consists of cells that are increasing in size. The increase in size happens through absorption of water or by osmosis and nutrients into the cells and vacuoles. Smaller vacuoles combine together to form a larger vacuole through a process called vacuolation. Water moves into the cell to create pressure against the cell wall, which then pushes, elongates, and widens the cell. The zone of cell differentiation consists of differentiate cells that differentiate once they have reached their maximum size. Differentiation forms permanent tissues such as epidermis, cortex, xylem, and phloem. The cells change their shapes and structures to become specialized cells with special function. There are two types of growth that occur in plants, which are primary growth and secondary growth. Primary growth is the growth that occurs after germination and it takes place in all plants to elongate their stems and roots. This growth takes place at the apical meristems at the shoot tips and root tips. Primary growth starts with active cell division at the apical meristems followed by cell elongation and differentiation. At the shoot tips, leaf primordial and shoot primordial will grow to form new leaves and shoots. This helps the plant to grow taller, while at the root tips, the root cap becomes exhausted when they penetrate the soil. Next, secondary growth occurs mostly in unicots and some monocots to increase the diameter of the stem and root. Non-woody plants such as herbaceous plants do not undergo secondary growth. Secondary growth results from the division of lateral meristem cells. Lateral meristems consist of vascular cambium and cork cambium.
However, plants can be classified based on their life cycles, which is divided into three, such as annual plants, biennial plants, and perennial plants. Annual plants have only one life cycle for a year. The plant grows from a seed and known as germination. Examples of annual plants are watermelon and corn. Second is biennial plants. It takes 2 years of growth seasons to complete life cycle. During the first growth season, the plant germinates and grows roots, stems, and leaves. During the second growth, the growth flowers and produces seeds. The plant will die eventually. The example of biennial plants are carrots and cabbage. Next is perennial plants. It lives more than 2 years and is classified into two categories, which is woody perennial plants and herbaceous perennial plants. These plants are able to adapt to their surroundings and temperature change due to their structure. Perennial plants do not die after 1 or 2 years. The example of perennial plants are mango and coconut trees. Moving on to growth curve for annual plants. It contains one sigmoid curve only. Stage A is decreasing dry mass. Food stored will be used for germination before leaves emerge to complete photosynthesis. Stage B, increasing dry mass. Rate of growth increased rapidly due to plant have carried out photosynthesis. Stage C, constant dry mass. The rate of growth is 0 and plant is matured. Stage D, decreasing dry mass, happens slowly due to lower rate of photosynthesis. The growth curve of biennial plants has two sigmoid curves which are combined. First growth season, plants produce leaves, photosynthesis takes place. Food is stored in tubers. In second growth season, food is stored and to produce flowers and seeds. Lastly is growth curve of perennial plants. It's a series of small sigmoid curves. Every year is a sigmoid curve. Growth occurs throughout the lifespan of plant. Highlight intensity increases the rate of photosynthesis. Rate of growth increases during spring and summer.
This is the end of our video. Thanks for watching.